Just the sheer number, if you kind of try to figure out how many of those things were on the seafloor per meter, you know, it was pretty spectacular.
For nearly 20 years on the bottom, that's quite a statement there, Chris.
Well, I'm not taking it back.
Virgin Ave, another 20 meters, 131.
That's my stance, and I'm sticking to it.
Chris has been active diving in submersibles since 2001, you said, right?
Yep.
That's all right.
Back then, you were in the submersible, though.
Yeah,
physically.
That's right.
Yeah, that must have been cool.
Yeah, it is, but so is this.
Yeah, this is super cool, too. It's super comfortable, also. We can go to the bathroom at will, which is nice.
So it sounds like we have some new viewers.
Okay.
And they're asking, what's that thing that you're sending down to see what you're showing? And also, how does kelp grow?
How does kelp grow?
Yeah.
Well, I can tell you one thing, kelp doesn't grow out here. Kelp's shallow water. It's along the northwest California coast. It's photosynthetic. It's kind of like a really fascinating algae. So it's growing up toward the sunlight. It's got floats on it to keep it up there and up near the surface, and that's what it's doing. It's a plant. And these kelp beds can be so dense that it's kind of tricky diving in them. I haven't dove in them myself, but what's that?
That was Trevor, I think.
Okay.
Yeah, and the second part of that question is the ROVs, right? So these are remotely operated vehicles. That was the question, right, Aphrodita? What are we doing?
Yes.
We're sending down? Yeah, so these are incredibly specialized and complex ROVs, remotely operated vehicles that we have on the ship, and we drop down to the bottom. There are two of them. The one you see primarily from the other view is Hercules, which is the primary vehicle. It does the collections. It has a high definition camera on it.
Bridge, another 20 meters, 131.
Has a 100,000 plus lumens of light to show the bottom. Has a manipulator arms to collect things, and it's all controlled from the ship, from a control room that we're sitting in at the top of the ship. And we have a couple of folks up here that are ROV pilots and navigators that communicate between the ship and the devices on the ROVs on the bottom. And there's a second ROV that's giving you this perspective on Hercules, and that one's called Argus. And that one, it buffers the Hercules from the ship, from the movement of the ship, and also allows us to monitor Hercules and get a perspective on it as well. And that has lights and cameras on it as well. So that's the short answer.
Yes.
I'm sure we could talk all day about the incredible technology that Ocean Exploration Trust has invested in and developed with this equipment. And lastly, we have the ship itself, which is a piece of wonder. Has cranes and such on it to move these really heavy machines around, these ROVs around. Whole crew and cooks and everything else to maintain us out here.
And just a few electronics in this room.
Just a little bit. We're surrounded by, I don't know, 100 monitors. I mean, I can't even count them all. That's incredible, though.
If the viewers go to the quad view, they can see the room full of computers.
Yeah, great.
Electronics.
Yeah, the quad view on your nautiluslive.org website. You can see our control room here, which they recently expanded. I didn't see the earlier control room, but this one is kind of 3 containers merged together. Behind me is a telepresence where they're communicating with classes. I think right now they're talking to a class at Hawaii Pacific University and interfacing with students there. So we've got quite a lot of things going on. We have a video engineer over in the corner who's collecting the imagery and processing some of that. It's quite an amazing operation. And on the website, again, there are great details about both of these vehicles, Hercules and Argus, on the website. So check that out and let us know if you have any other questions.
Yeah, thank you for that question, Trell. He's 5 years old.
Nice.
Who asked about the kelp.
Future ocean explorer.
And also for our new viewers, if you follow us at nautiluslive.org and the main page, you can see more information about where our rovers are. So right now, if you look, you can see Hercules at 1,734 meters deep.
Virgin Ave, another 20 meters,
And
131.
Argus is just a little above Hercules. And you can see also the water temperature is 2.3 degrees centigrade.
Ooh, chilly.
Very.
Just above freezing.
So there's lots of information in our website. So thank you for those questions, and we'll continue answering them.
And again, we are operating in what I like to call the largest fully protected conservation area on the planet, Papahanaumokuakea Marine National Monument. And we just recently initiated a process to consider this area. NOAA is considering this area as a National Marine Sanctuary. And so we would like your input on that process. We're going to have 4 public virtual meetings in December. And also we're collecting public comment through January 31st. So please go visit our website, papahanaumokuakea.gov, and let us know what you think. If you want more protections for this area, that's what the proposal is doing, more and lasting protections for Papahanaumokuakea. It protects some of these amazing deep sea habitats and incredible coral reef habitats in the shallows, and a lot of the things that we don't even know because we haven't even explored much of it.
There's a fish, I think. It's in the left top or middle thing. It's very small.
Where is it?
It's to the left of lasers. Keep going, keep going.
Lasers.
It's very small.
Let's take a look if we can figure out where it is.
Might be the end of our leash. Where are we looking?
It's to just
there?
a little bit left and up from the lasers.
Left and up.
Oh, I do see it. Okay.
What
Yeah.
are you guys looking at? This is
Go ahead.
crazy. I see nothing.
Drive a little bit ahead.
And for the
I've lost
viewer
it now.
who's asking,
I still see it. It's just about to go out of the top.
ooh,
Yeah, you're gonna have to go forward there, Antonella.
the lasers are 10 centimeters apart.
Left a little bit more,
And
and then
then
straight up now.
Yeah.
Oh,
Almost
I
coming
see
into
it.
between the
Where?
lasers,
You see
right
it, Chris?
above
That
that.
Right.
tiny fish.
Right in the center now.
Okay, there we go. Jeez. Good.
So
I'm getting
Jeannie.
old.
Oh.
The rat tail.
Yeah, this is going to be Kumba. How about that?
Yeah,
Oh,
we saw one of
Kumba.
those
I
earlier.
like that name. Kumba.
Kumba.
We saw one.
Yeah, this
And,
is in the family
morning.
Macruridae. It's a really tiny rat tail. That's why I could go to that. And it's also very dark colored.
Yeah. You could see the shadows from all the
Yeah.
lights from Hercules, multiple lights shining on it.
Do you have binocular glasses on over there, Aaron? the
No.
videographer? What on earth?
It is impressive.
I used to think I had pretty good eyes, but holy guacamole, she's got serious eyes.
When we ask for a zoom on Herc, we just use her eyes.
Virgin Ave, another 20 meters, 131.
Yeah, Aaron has a lot of experience of observing the wild and
By the
Filming
way,
the
excuse
wild.
me, if you look left, just slightly, about halfway over to up the panel on the left, there's some type of a coral or something, and it's about to go
Can
away.
you telestrator it?
It's gone. It was the lower left corner. It was gone already.
Okay, if it's really important, we'll go back. Otherwise, we'd like to move along.
Let's move along. Not
Okay.
that important. Pretty sure it was a
Roger.
black coral, but
And in the science chat, so when we mentioned the science chat, we have a special portal that allows experts from around the world to watch what we're looking at and provide comment and provide extra identification and other information. And some of the information we mentioned before from Kenneth Sulak, and he is telling us now about the Chimera, which is really interesting. So in this environment, it's pitch black, and these Chimera have this special adaptation, the ampullae of Lorenzii, which some sharks also have. These are modified lateral line pit organs, and they're electroreceptors, so they allow it to detect other fish movement and kind of like a metal detector.
That's the end of your leash.
And they're detecting the weak electric fields generated by other organisms. And sometimes they can do that to detect organisms beneath the surface or just other fish around them. And it allows them to detect left or right. He says deflections in the electric field alerting to the location of the prey. So that's a really cool adaptation to have for a fish in this environment. Oh, is that an anemone you're circling there, Chris?
Do you want to zoom in on that?
Sure, if we can. We haven't seen too many of these on this cruise. This is what's called a hormathiid or Venus flytrap anemone. And that's as far as I think the actual genus now is known as Philiactis, and can't go any further because the rest of the taxonomy is all based on internal structure, which we cannot see.
So, didn't put on the X-ray cam.
Yeah, the X-ray cam, even that, it's soft mesenteries inside of it.
Oh, wow, that's beautiful.
They're really easy to spot if you see the ring of tentacles right around the lip of it. Then there's a clear space, and then there's a very sort of horny cuticle type of stalk on it. It's very distinctive, so it's a very obvious pattern. Thank you very much, Antonella. That's a great shot. And look at how big that thing is. That's a whopper.
20 centimeters
20
across,
centimeters.
roughly.
Regina, another 20 meters, 131.
So a fact about the ROVs, although they share some letters with rover, a rover means it's a vehicle used to explore extraterrestrial planets. So planets or moons or something like that. That's where we use, you've heard of that in the Mars missions or the lunar rover. This is very related. It feels like outer space, but it just happens to share a lot of the same letters. ROV is actually an acronym for remotely operated vehicle. So different than a submarine or a submersible. These are remotely controlled from the ship. And yeah, ROV Hercules and ROV Argus are both controlled by us up in the control van.
They used to refer to submersibles as HOVs, or human-operated vehicles, but then that's confusing because this vehicle is being operated by humans too.
Right,
They're just not in it. But also, I think it also is meant to say human-occupied vehicles.
Which conveniently shares an acronym with high occupancy vehicle. So you can use a submersible in one of those lanes.
Going down a slippery slope here.
Now, if we used an ROV on a watery moon, an ocean moon, I think then it would be a rover ROV, which would be pretty cool. Maybe one day we'll see that. Hopefully in my lifetime.
Yeah. Well, we're fortunate enough right now. I mean, I'm older, so I don't know what I'm going to have left to see, but I'm very fortunate to see what's going on with Mars as it is right now. Who on Earth would have thought that we would fly a helicopter on Mars?
Yeah.
Yeah, that's pretty incredible.
So we climbed up a bit of a wall, and then we're sort of onto another sort of flat area for a little while, so the nodules have reappeared. Those of you who are following us from this morning knows this is quite a bit different than what we were seeing when we started the dive, which was spectacular, dense coral and sponge communities down there. If you're just joining us and think this is a boring dive, you're going to need to go onto YouTube and look at the video of the earlier part of this dive, because it was just something else. And what we're doing right now is we extended the dive because we want to really try to make sure we understand where this ends, the very top limit of it, because we are considering a second dive on this community going below where we started last time to see if we can find the lower limit of this. This is really valuable information. And so we're not going to do that tomorrow because we're going to have to run with our tails between our legs. There's a weather system, sea state system, that's going to sort of have us aboard our ROV operations just for a day or so. And we're going to shoot south and take advantage of that time to map some seamounts that we're going to dive on later on in this cruise. And then we're going to come back up and resume. So that's the plan.
It is winter out here in Hawaii, and the same weather systems that generate the massive swells
20 meters 131
that impact the north shore of Oahu and create those incredible waves of Pipeline and Jaws are also creating swell that crosses across our path out here. So although they are able to operate the ROV and even do mapping up to a certain swell point, if the swell is too much, they are unable to operate. And what is that point, Chris, or ROV team?
What point?
The point for swells.
There's a fish
like
to our left. It's just going off screen now.
It's hard to answer that in a one-stop shop answer because there's a lot of other things that come into play. If there's absolutely zero current, zero wind, and zero other weather problems, then we could get away with a lot of swell, but again, it depends on the period of the swell. So it's not just the size. If you have really long period large waves, you can get away with it. But really short period large waves, that's not good either. So it's more of a, I don't know, more of a qualitative weather judgment than quantitative.
Are you happy for me to zoom?
That period
Are you
he's
ready for a
talking
zoom?
about is the steepness of the wave.
Give me a second when it gets back to center.
A long period swell is kind of like a gentle, it may be large, but it could be a gentle slope. Oh, this is this one that was doing that weird mouth thing before. Occasionally stops and shakes its head. Was this the same one we saw before, Chris?
Ooh.
Yeah, I think that's the head shake, I think, is a response to either the ROV noise or the light, probably the light. He just doesn't understand it. I almost wonder if when he eats certain things, some things fluoresce as sort of defensive mechanism, and he almost thinks as though something like that's going on. I don't know.
Something that can. It's really interesting how so many of the fish down in this deeper part of the ocean take that eel-like form. Maybe Kenneth has input on that as to why that's such an adaptive feature down
another
here.
20 meters 131.
I'm getting pulled. I can come down a little bit, I suppose. I guess we're going down slope.
So Trevor, what are you driving right now? Hercules or Argus?
I'm sitting in the Argus seat.
Oh, so that means that Antonella is driving
Flying Hercules, yes.
Hercules.
Well, unless Trevor has done something interesting to the back of his hair. Yeah, it's Antonella up there.
I've actually developed a wireless remote control.
Yeah. Our viewers are curious who's driving each of our ROVs.
We like to switch the seats up every now and then to do some cross training and get more experience in different areas.
Sure seems practical to me.
Not to mention she's doing a great job.
Yes, absolutely.
So for our viewers, Trell, If you and your aunt want to go to nautiluslive.org on the main page, you can see how deep the ROVs are. So you can also see who is on watch, who is videoing what we're seeing and taking pictures, who's the navigator. You can see the whole team every 4 hours.
The website
The
also
team
has
changes.
some really great resources where you can look at people's profiles and see like what their education was, what they've done before. And then also it has little breakdown videos or explanations of what each job does. So it's a really great resource. And sometimes they have
Yes.
internships as well. We don't have as many this year, but I think they're going to end up doing internships again.
I let out a little chuckle earlier. We're sort of past it already, but the lasers are on that big rock, and it reminded me of that sci-fi movie. And for the life of me, I can't remember what it was of the brain bug that it was right at the end of the movie. They captured it. It was anyway. I can't remember what it was, but lasers looked just like the eyes, and then the shape of the rock was just like the brain bug, which was the alien. So anyway, okay. This must be the second watch of the day, huh?
So we have a question here from a university student who was studying the tree of life and learning more about sponges. He's asking how sponges and other organisms have adapted to the cold water temperatures this deep.
What we consider cold is not necessarily cold to all of these animals. It's not really clear where sponges evolved, but it wouldn't surprise me at all if they actually evolved in cold, deeper water, and then eventually there were some species that managed to get up shallower, but I'm not sure about that.
Bridge nav 20 meters 131.
It's a question you can't really answer because you don't. I don't really know where or what depth they actually arose from.
They're definitely one of the more successful groups in the animal kingdom, one of the oldest groups.
Yeah.
Also.
And one of the simplest animals as well.
There's the lesson right there. Sometimes the simplest is the best solution.
And have the longest lifespan, would you say?
I'm not sure.
Hard to say because we haven't been able to measure that in sponges. There is some measurement of some of the corals, specific types of corals, and there are actually some individual colonies that they believe have been alive for thousands of years. And it's only specific types of corals. It's not all of them. And it wouldn't surprise me at all if eventually when they figure out how to age sponges, where they're going to find some species of sponges can grow to be that old as well.
So a viewer asked maybe like 10 minutes ago, what are some cool facts about sponges and corals?
Okay, well, I'll start with sponges. Some people sort of think, well, they're just sitting there. They're not moving. They're not wagging anything. And that doesn't matter. I used to be a fish guy, believe it or not. And I still love fish, but then I've found sponges to be absolutely fascinating. And for me personally, it's their architecture. Their taxonomy is still in development. We're not anywhere near understanding all the taxonomic relationships and how different type of sponges are related to each other. But and also the taxonomy is now changing to more of a genetics-based field, or if you will, and it's always traditionally been morphology and based on microscopic parts of their bodies that are called spicules. And the way you traditionally have tried to identify a sponge is you collect the piece, you take a piece of the sponge, and then you can put it into something as simple as household bleach, and you leave it in there for a little while, and the bleach actually dissolves away the tissue of the sponge, and all the little microscopic silica spicules which give it some support and structure, they are then left. And because the bleach does not dissolve those spicules, and you take them and you put them under a microscope, and they're all different shapes, they're all different sizes, and it's which spicules are present in which species is how you actually try to identify what family and what group of sponge it's in. In some cases, there are sponges that all of their spicules are loose within their tissue, and they're the ones that are kind of like a kitchen sponge, you know, where you can grab them and they'll compress and then expand and compress and expand. That's a bunch of sponges. A lot of sponges are like that. And then there's also a lot of sponges that instead they've secreted additional silica to actually glue their spicules together to create a very hard framework, which is referred to as a dictyonal framework. And this can take many shapes, and you know, people study these frameworks where, you know, what's connected to what. And so that's the answer about sponges. Their structure is what I like. I like how they actually attach to the bottom. There's various different mechanisms. The way they filter, they actually can create their own current.
Okay.
And we're looking at one of these spectacular sponges right now. This is
That's all I got.
called Poliopogon.
Oh, it's gorgeous.
And there were tons of these earlier in the dive all over the seafloor. And that's
oh, that's a nice view of how it's attached.
yeah, and this is called lophophytus attachment. It's actually grappling the substrate. It's not gluing to it. And that seems to be a very successful way of holding station in strong current flow. Corals, I don't know what to say about corals except they're absolutely beautiful. They're all different types, all different shapes. Various types of shapes and sizes, just like sponges, are successful. And they're all filter feeding animals. They're getting nutrients and organics and catching things from the water column. So, but sponges again are more like trees, I guess, if you will. They have all types of branches, and both of these types of animals are attempting to expose as much surface area to the current so they can catch as many things as possible. I don't know, I hope that answers the question. That was kind of long, but
that's awesome.
Thank you. That's helping me too. I'm writing my notes.
yeah, I'll try to be a little shorter next time.
And if you go on the Googleplex and you type in sponge spicules, that's S-P-I-C-U-L-E-S, sponge spicules, you can see some really cool high-resolution images of the different forms that these glass, these sponges spicules take. It's almost infinite. It's beautiful.
So that potbelly sponge is called Hyalonema, subgenus Corynonema, and we discovered that thing in 2015. It still hasn't been described.
Nice movements for the video, Antonella.
Oh.
Yeah, now I guess we haven't found the end of the community because they're starting to pick up as we come uphill.
Quite a bit.
Yeah.
So what's the pressure?
If we do, we should get closer to him. It's that.
We're good for now.
Yeah, what's the pressure at this depths of the ocean?
Okay.
Oh boy, it's about the same pressure.
That was a really nice
if you were
pull-out,
able to
Antonella.
crawl inside a scuba tank, a 3,000 PSI scuba tank, it would be about the same pressure.
Wow.
You could have an African elephant stepping on a square inch of surface. That give you a visual idea of how intense the pressure can be.
Is an African elephant 3,000 pounds?
Yeah, I think about 3,000
That's a large
pounds.
animal.
It is. It would be hard for it to step on a square inch.
Yeah, only when it wears high heels.
But yes. Well, we are at, what is our depth right here? We are at 1779.
What? No, 1720.
1720.
Yeah. I think at least in my display.
I learned that stellar sea lions, the ones that are up in Alaska, can weigh up to 2,500 pounds.
Wow.
Pardon me? Say that again.
Oh.
What?
The stellar sea lions, like the ones up in Alaska,
Oh, sea lions.
sea lions can weigh up to 2,500 pounds.
That's also a large animal.
So why aren't they called sea elephants?
We'll have to get on that and tell them.
It would be my recommendation, that's all. I think they're lions because they're predators, aren't they, Aaron? I think you might have been up there filming some of them.
Yeah, I have filmed them. They are, but we were filming them getting predated on, so it wasn't really them predating at the moment.
I see.
The orcas were going after them.
Oh, I'm not sure very many African savanna animals go after lions.
Oh, for lions. I thought you said sea lions. Now I'm all mixed up.
No, I was saying changing the name, right, to sea elephants. And I was thinking that maybe that's because these sea lions are predators, but then you were saying in your case you were seeing them being predated on.
Yeah, I think they eat a lot of fish, and probably, I think I've seen them eating like an octopus, but I could be wrong.
So
Have you filmed lions?
I did, yeah, I have.
that is a echinotherid urchin right at the edge of the, I'll do a little drawing here. They imaged one earlier. And by the way, this ledge is pretty interesting.
Yeah. This is
I'm hard pressed to believe that's all crust, but it could be. That's really thick.
Oh, what is that thing right there? Oh, it looks like a...
So this is a sea urchin, and it's in a family that's called Echinotheridae, and it's very...
Yeah, we got a lot more zoom.
It's very poisonous. The genus is Sparosoma, and if you grab one of those things with your hands, you're going to regret it for a long time. Your hands will swell up.
Oh, what's
And
in
you've
that,
done
in
it
that little
before.
Bridge
cave?
nav, 20
Want me to
meters,
go wide,
131.
I have not done it myself, but I was with somebody who did it, and we were both ignorant, and she just happened to get to that animal before I did, and I felt very sorry for her for a number of days, because it was a very painful experience.
We have another question here. How do sponges digest their food?
Dinner release here. Hurry back. I'll miss you guys.
That's a good question. I don't know a whole lot about the physiology of sponges, but remember, this is a very simple animal. They don't really have mouths, and they don't really have digestive tracts or intestines or anything like that. So a lot of the energy they get from their food is at a cellular level, and so they're filtering extremely small, presumably organic particles that are captured in the interior of the sponge and then somehow absorbed probably by individual cells. I'm afraid that's the best answer I can give you. I don't know anything more than that. And they trap these particles, and then the water afterward just shoots out the other side of the sponge. So it's a continuous one-way flow.
Thank you. Our viewers are very grateful for all these explanations.
Even though they're a little shaky, because that is, okay, all right.
Well, getting back to the pressure calculation.
We're
So
doing
every
131,
10 meters,
20 meter steps
you
up to the
get
top.
an extra 14.27.
What's that?
We're doing 131, 20 meter steps to the top at the moment.
14.27 pounds of pressure per square inch. So where we are now is actually not quite an African elephant. It's only half of an African elephant, but 2,454 pounds per square inch, which is still a lot.
Bridge nav, another 20 meters, 131.
came just in time for the rocks.
Oh, you've collected all the ones you can collect today? Your rocks you have collected. Wow, aren't you excited?
Don't we have some saddlebags on the side? We can just toss them in there.
Oh. Okay, viewers, we're making a few little changes here. Some of us need to get some dinner, so we'll be right back, some of us.
Yeah, so getting back to the sponge and these currents that it generates, there's a really cool video online where somebody actually injected some dye into the area around a sponge and videoed how it moves through the sponge. So definitely look that up online. It's really fascinating. You get an idea for how complex these organisms are and how the currents move through
crank
the
it up
sponge.
if you want. Might find something cool to look at though.
Oh.
You're good.
All right, I'm going to
Too many buttons.
get something to eat.
I want to see that video where they put dye into the water and goes through a sponge. It's on YouTube.
YouTube. I always forget to bring a coat. Ooh. Freezing.
What is
So
that
Charlie
pink
Brown
thing?
coral.
Is it Charlie Brown coral, the pink thing?
Why?
Looks like it has some barnacles in it. Does it?
I don't hear you so well.
Can't hear me?
Just a little lonely coral there.
How about now?
That's better.
Bueller?
Do you
That
guys
looks
want
like a bubblegum coral, no?
I don't know. Zoom in. Zoomy zoom.
How good are you on coral identification?
Not very. I just say anything that's like curled up like that, it's a bubblegum coral. I'm like, I'm sure I'll get everybody telling me, no.
Zoom in again if
Yeah,
you
but
want.
you could tell us everything you know about rocks.
Yeah. Everything I know in one sentence. It does look like
Pretty.
the polyps are not, unless it closed up, unless they closed up.
It's pretty though. It's a nice
Yeah.
color.
And the brittle star is pretty too.
I'm going to give you an upvote. That's a pretty good guess.
You think you already have a good guess?
No, I think that you're...
Oh,
I
my
think you're on.
bubblegum coral? Oh, well.
How about this one?
How about this one?
Plexard? Maybe, no, that's not, unless it's all closed up.
So yeah, that's the thing. We don't know if it's all closed up or that's the way it's open, right?
Right, let's see here. Compare this to the hemicoralia. Yeah, it's a good enough zoom though.
Yeah.
Okay, moving on.
Paragorgia. Sure looks like it.
Yeah. Does look like it.
Everything looks like Paragorgia to me.
Oh yeah, you already said that, Dan, that everything looks like a bubblegum coral to you.
Pretty much.
Pretty much.
Is that a tube anemone again? Bottom right?
Bottom right.
Bottom right.
Keep looking down.
Still below me?
Oh,
Yep,
I
right
see
there.
it.
Oh, yeah. Yeah. Can zoom in there a bit.
Good eye.
Yeah, he's a little bit different color than the last one we saw, huh?
Yeah.
Not as dark.
Mm-hmm. So we saw 2 of those on our watch, and then here's a 3rd one. So at this higher elevation, they are...
It doesn't have that like inner ring of stuff that the other one did.
well, yeah,
Zoom in
maybe
a
it's
bit
because
more
we're
if you
not
want.
zoomed in, or maybe I don't know. But that was the primary question that the specialist had us look for, the internal tentacles.
Okay.
Yes, thank you.
Maybe we should slow down again. Getting
Okay.
run over.
Bridge nav.
Lots of stuff up here to look at.
Can we reduce that speed to 0.2 knots? And let's reduce the step to 20 meters. Thank you.
Big sponge on the left or colony on the right? Argus view.
Oh, well, look, there's a coral right in front of us.
Oh, I don't mind.
That's taken us in the direction of the coral colony.
Kind of come around to look uphill here.
We have a viewer wondering why aren't there that many
it's
fish?
on a movable rock. Okay, you can zoom in there.
Not that many fish. We're getting that question
See
a lot.
few, but not.
Yeah, and in this area, what was a
Couple things going on. Allogatrophic
alogotrophic?
waters. It's not a lot of snow raining down, so we don't have a big buildup of
Bridge
sediment.
nav, hold position.
So a lot of fish would prefer the invertebrates that live in the sediment for food. But we do get some scavengers, like that chimera, was ghost shark, was really wild.
Yeah, and we have eels.
Right.
Probably more scavenger type organisms, right?
Yep.
That's a beautiful coral.
Yeah, not a lot of nutrients raining down, and it's a very rocky bottom. Not a big accumulation of sediment.
Okay.
Yeah, thank you.
I'm lost. I changed my heading. I think I wanted to go look left again. There was a big, I think he was a Dr. Seuss.
Well, there's
maybe.
another CNN guys right there. Either look like
several.
You want to see those guys?
Yeah, I think they might be again those tube anemones.
It's more like the color that we saw earlier.
Zoom in there if you want. Yeah, you'll have to push right in. It's close up.
They look different.
They do look a little bit different. The question is, do the two look the same that are right in front of us? I don't know.
It's got kind of a big stalk there.
Yeah, he does.
So yeah, the tube anemones can have tube can extend 1 meter down into the sediment for some species.
Wow.
here, I don't think you'd get that deep a buildup. It'd have to be like horizontal.
And Sarah asked a very astute question and said, well, can it go sideways? And yes,
Right.
it can.
Okay. Yeah, I see him there. Not sure which way to go to get there.
There's a viewer asking.
So I gotta go 180. Is that what you're saying?
Tell the criteria. Can you tell the viewers what the criteria is to determine if a coral community is healthy? Like, what do you look for?
I think we look for diversity, and that we don't have a lot of, like, every once in a while we'll come upon a community and there'll be like a whole bunch of downed corals or a whole bunch of downed sponges that you could see there was a community there before, but it's not very abundant, you know, now. I think that's one of the things you look for, but I think diversity is, right, one of the things we look for a lot.
Well, that's a healthy community. You know, it's a diverse biodiversity. It means more resilience. But for the individual corals to tell whether they're healthy or not, I think you want to look and see if they've been preyed upon, see if the tissue looks intact.
That's funny.
You're not seeing skeleton, but the polyps.
Oh yeah, that might be another branching bamboo coral. Remember the one that had the...
Yeah, the undefined.
Trident.
Yeah, this is maybe not that. Let's see.
If it's all in one stalk, it might
looks
be a
like
giant trident. Yeah.
Well, the trident, you would have one stalk diverging into 3.
I think
and
it
then
is.
from there outward.
We just can't see it yet.
This looks like a primnoid, maybe. Well, oh, you might be. Look at that. Let's see.
So this one is not well described, so we want to get as many images, if it is the one of those, as we can.
The one that we saw was a bamboo.
Yeah.
This is bamboo. You can see it on the stalk.
Yeah. That could be.
Now, will you drop a point in HiPAC?
Sure.
Thank you.
And if we could zoom on where the branches come off of the main stalk.
Around it.
So there's not much known about these, or it's not been seen
Not
a lot?
much. They haven't been completely classified or described. So we ran upon one in our last watch, and they were really excited, and we got a view for them. And so this trident shape of the branches, so this is a branching bamboo coral, but also this trident shape is indicative of them. So he wanted to get a view of the center and of the polyps.
That looks like it. So it starts out with 3, branching into 3, but then you get pairs coming off the older it gets.
Yeah, so this one is also, he said, so the one we saw previously was very old and healthy, and this one looks, if the identification was how many new stalks you have from the trident, so this one looks even older than the one we saw previously.
Wow.
Now you can call it a trident coral or a
It was
candelabra.
a branching bamboo coral.
Yeah, branching bamboo coral.
So our sighting enabled them to describe it as being in the North Pacific now, as well as the North Atlantic.
Right. That's correct. That was one of the big finds of our last shift. He was really excited.
It's really pretty.
It is. I love it. I was wondering, in the other time that it's seen, is it also in high-density sponge communities? That would be interesting to know.
Could we zoom out, get the whole thing in the frame with no lasers?
Sure.
Or one with, one without? Wow.
Beautiful. Beautiful.
All right.
Happy?
Yep.
Yes, thank you so much.
Off we go.
One that size, how old do you expect it to be? I know you can't really tell, but...
He really, Scott France said he didn't have a clue.
Oh, great.
Yeah.
It's just relatively older than others, the
That
more branching you get.
Yeah. And I think it was Steve's talk, right, where he said a small branched coral that we saw was, and it was small, like maybe a foot, maybe a half meter, not even.
Oh, the stony corals, they grow very
corals.
slowly, and so that one, yeah, was about 60.
Yeah.
It was an old one, and it was very small. So if you see a large one of those, you're talking hundreds or thousands of years old.
It reminds me, I gotta rake the lawn when I get home.
It is pretty. Yeah, I agree.
I thought that might be one of your mystery sponges, but I guess not.
Okay, I'm back.
You had a
Very
quick
good
dinner.
Yeah, it's a very good dinner. So it looks like the population of animals is picking up. Not quite the way it was this morning, but there's still it's a decent community up here.
Yeah.
Am I high enough to clear that thing? Looks like it.
Okay, ready for a move?
Let me come back around here and yeah, I guess so, yeah.
Okay. Back to heading uphill. Okay.
I see there are not very many of them. It's just a little cluster here, it looks like.
Seems really common to have those kind of really sparse and then just these little patches of them.
Yeah, the substrate's a little bit, they're consolidated patches mixed in with unconsolidated patches, and you can see the sponges sort of clustered on the harder, more durable substrate.
Just zip over here to this little rock.
Yeah, that's a little more extensive over here.
Have a look around. Might want to hold here for a minute.
Yeah, I didn't call anything in yet.
I see we've got an Iridogorgia. I don't remember seeing one of those down below. That's that very woolly sponge over there on the left-hand side.
Chimney cleaner? Oh, no.
Chimney cleaner or bottle brush.
Yeah.
Beautiful sponges. Probably one of the most beautiful down here. And then we got some more of various types of massive sponges here. This is a little bit more like the whole substrate was like down below.
For if you were wondering if they can find a dive schedule, we don't really have one. We have one tentatively for each expedition, but that does change and it varies. So your best way to know when we will dive is by following our social media pages, Facebook, Twitter, and Instagram. And we do always post dive alerts when the ROVs go in the water. But we don't have a set example 9 to 5 or a set schedule of dives. Everything fluctuates on weather, what the ship is doing, how the ROVs are. So we just kind of figure it out as the expedition goes. We do have a set number of dives that we hope to achieve, but the start times can vary depending on the conditions.
He's got some big feet there, big hairy feet.
Kind of wonder how a sponge that size is so successfully clinging to a vertical face of this boulder, or this, it's not a boulder, it's a block, I guess is what you'd call it.
Do sponges grow at the same speed as corals?
Yeah, that's a good question. I doubt it, but then I have no information to back that up. I don't know, it's a very good question. I don't think a lot of people know how fast these deep sea corals grow, and we definitely don't know how fast the sponges grow. The only thing I can say is that we've seen both sponges and corals on wrecks, and we know when the wrecks actually got into the water, so that we know this is the maximum amount of time that they had to get to that particular size. But we have no idea when they actually settled on the wreck, so we can't get a good estimate. This is pretty. Look at this cluster.
Some of these sponges are just so large that you would think they're quite old. But again, if we don't know.
To go all the way around that rock.
So you told the viewers that we're now exploring the planet Venus, didn't you? You didn't tell them that?
I did not, but you just said.
Those of us who've been doing this for a while, we see these scenes, and that's kind of what we think. We think perhaps this is what an alien planet that can support life might look like, because this is so completely different to things that we're used to seeing.
Looks like it.
Yeah. A little coral down here.
Which coral is that?
I think it's a primnoid, but I wonder if we could do a snap zoom on that guy right there.
Yeah, I can zoom in their video.
Standing by for you guys to tell me. That's what you'd like? 10 meters, and what was the bearing? 135, sure.
Aaron, are you there?
Getting weird.
Yep.
Can you zoom in?
I can.
Bridge nav.
That's good, thanks.
That's pretty.
Yep.
Can we get a 10 meter step 135?
So this is another beautiful bamboo coral, and presumably it's got a little bit of an odd shape because of the current flow. It's responding to the current flow, I think, in the way it grows. Bamboos are magnificent, and they also host, there's a little cup coral right above it. I wonder if we can.
First cup coral I've seen.
First cup coral of the cruise, I think.
Totally.
Really?
See where I am here. Okay, push in a bit more, Aaron. Thank you.
We were wondering why we weren't seeing more of those. And you're probably wondering why we're getting excited about the single polyp little coral when we're looking at a beautiful big one. But we've seen a lot of the big ones on this cruise, and we haven't seen very many of these little ones.
I haven't seen any of them.
Yeah, well, yeah, this is it. So this is actually a hard coral. This is related to the corals out on the coral reefs in, Hanoa Bay or something on Oahu, or some tropical area as well. But down here, these same hard corals that are called scleractinians, they tend to be forming single polyps in very small colonies here.
And are there stalked varieties and non-stalked varieties?
And the guy in the Smithsonian, Dr. Stephen Cairns, is an expert on these as well as a lot of different corals, and these are very complex to me. I don't know how to identify them. It has to do with the septa, how many septa are in there, in what order, and their counts on these, and the tentacles and the stalk. But that was good. We got a cup coral finally.
Yes.
Thank you very much,
Can we get
pilot.
another